This is a special episode of the Stem Cell Podcast, ISSCR 2025, Day 3. Hello, everyone. We are Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. We're back with our third episode covering highlights from the ISSCR 2025 annual meeting in Hong Kong. Arun and I have had so much fun attending and getting to meet all of you, but it's not over yet. If you're looking to find out what we're up to at the meeting, follow us on X at Stem Cell Podcast or on Blue Sky at STEMCELLPodcast.com. You can also find episodes covering the 2 previous days of the meeting at www.STEMCELLPodcast.com or wherever you find your podcasts. Today, we're going to be recapping Day 3 of the ISSCR 2025 annual meeting. We're going to kick things off in just a minute, but before we get to that,
are you looking to sharpen your skills in stem cell culture? Get access to free on-demand training courses to help you master techniques in HPSC, hematopoietic, organoid, neural, and other specialized cell types at stemcell.com/on-demand-training. I'm going to start things off with just a short recap of the presidential reception last night. Really fun. That was Thursday evening. Really fun event hosted by the president, of course, Valentina Greco, the outgoing president, I suppose, at a beautiful venue called the Watermark. It was like a glass building where, everybody who's who in the stem cell world got together. We're schmoozing, mingling, congratulating each other on a great meeting. It was fun to catch up with some familiar faces, including Sergio Pasca, who was in town. I hadn't seen him before the event, so it was good to catch up with him for a little bit, as well as Rekha Iyer, who was, as part of our NICEF episode not too long ago, a few months ago. Ming Shagu as well, who's at UCLA now. So great to catch up with old friends and colleagues and, guests of the podcast who have been on the show in various times over the last years. So bringing us to this morning, back to the scientific sessions, I went to a concurrent titled Clinical Translation of Bioengineered Tissue-Based Therapeutics, and this was led off by Ryuichi Okamoto. These were some really exciting, clinically oriented talks that were using organoid models and other tissue-engineered approaches for various clinical applications for, treating different types of diseases. Dr. Okamoto was focusing on clinical application of patient-derived organoids for treating inflammatory bowel disease, something that's really a universal problem that he was able to establish. It was this group, a sustainable organoid system with a type 1 collagen culture that can recapitulate the inflammatory environment of ulcerative colitis, which is pretty cool. And they did a xenotransplant system into a mouse model, had a really great quantitative longitudinal imaging monitoring system for the organoid culture. And like I said, had an autologous organoid transplantation approach. That was awesome to see. Somebody else who I'm pretty familiar with, it's Kevin Costa from Madeira. I believe he was at Mount Sinai not too long ago in New York, but he's a cardiac biologist focusing on a bunch of different things, honestly. But his work was in this presentation talking about a gene therapy for heart failure informed by stem cell-derived engineered heart tissues. This is a treatment for heart failure with preserved ejection fraction or HFpEF, something that's still a mystery in the cardiac field. Typically you think of heart failure, you have reduced ejection fraction, the ability of the heart to pump, but some forms of heart failure have actually a preserved ejection fraction. So they're developing an AAVS1 circa 2A treatment, which is actually already FDA accepted based on the FDA Modernization Act 2.0, which is really cool. This is alternative to animal models that are being used to push the study along, which actually help facilitate the FDA green light there. That's really cool to see. Next up, Wei Zhou from the Tongji Stem Cell Center here in China, using lung basal stem cells to repair the human lung. This is a great model for intrapulmonary transplantation of human airway basal stem cells into immunodeficient humanized mice. The goal here is to, of course, regenerate the lung in certain diseases such as pulmonary fibrosis and so on. Airway basal stem cell transplantation, which is able to actually regenerate the gas exchange units. And like I said, applications for COPD, idiopathic pulmonary fibrosis, a lot of different types of diseases of the lung. Xinzhong Li from Yonsei University, preclinical study of iPSC-derived ECs for peripheral artery disease. Again, another major unmet medical need. Looking at the integration of the ECs into vessels and their functionality, which is really cool, and improvements that were shown in a hind limb ischemia model in mice. And the exciting thing about this was that apparently it's one of the first approved clinical trials, IPS clinical trials in Korea, in South Korea. That's hard to believe, I guess, in a way, but iPSC trials are happening around the world now. And then wrap things up at that session with Marcelo Revolta from the U Sheffield, was looking at a pluripotent stem cell derived therapy for hearing loss. It's creating these auditory otic progenitors. I think we've actually talked about this a bit on the show, which are able to restore functional connections from the cochlea to the brainstem. One thing that was interesting about this system was that he was using a gerbil model, and I haven't really heard too much about gerbil models. Usually it's rodent models for these sort of things, even on the ear side of things. But apparently gerbils have a hearing range that is closer to humans, and I have no idea how you can figure that out. But yeah, gerbil model. So really nice preclinical results showing some hearing restoration with the system. And this Rincell 1 is the name of their therapeutic, when the goal is to be an adjunct to the standard cochlear transplantation system that's being worked on. So really unique clinical trial there.
For me, I saw his name in the program, and it was like gravity. Jacob Hanna, he went off in this Fate, Choices, and Development and Regeneration session of the concurrence. And, he totally has me conditioned to expect some kind of shocking news. It wasn't crazy, but he did extend. He shared some unpublished work, which I was hoping for and was delighted to see. And what it amounted to was, in just the first aim, he kind of blew me away with he had 3 kind of bench milestones that he'd met since you'd last heard from him, which is like 6 weeks ago, it feels like. But in the first, he showed how he extended this ex utero embryo culture device to embryonic day 13.0 equivalent, which is, may not sound like a lot. I think he had it to 11.5 was the limit before or something, but that it's a lot happens in those days. And what he showed is that the more organ development, more refined organ development, and little things like the digit formation and the limb bud and like the eye forming, like things that are real and important in terms of the clinical translation, also showed that the system as they had, and there were some tweaks that he made that he announced that he was going to be dropping any day now. And he's teamed up with Oko Lab to make these ex utero culture devices commercially available. So it's it's not going to be cheap, I'm sure, but entry point for anyone to do these kind of studies. And a key distinction he showed is that these systems don't just work for like egg cylinder type development, which is what the mouse is. He used rabbit embryo, which is a bilaminar disc, much like humans. It showed that those would develop to gastrular stages as well, ex utero. So the implication there being, of course, that human embryo development or non-human primate embryo development would be feasible in this ex vivo culture system. Although the timeline's obviously going to be different, maybe the scales, something that needs to be worked out. And I'm sure he would be the first to concede. But point being is that there's an incremental advance in the efficiency and capability of this device that I think, the latency being what it is in the scientific realm these days is not very long. I think we can expect to see a lot of scientific questions being addressed across many different labs beyond the Hanna lab toward that point. Also, I think maybe the more impactful announcement that really had the audience stirred up was his description of what he's calling alternative conditions media, which was based on Han Qi-Dang chemical formulations. All the work that Han Qi-Dang has done over the years was kind of the foundation for this cocktail. So Dr. Deng's name ringing out again. I think he's probably the MVP of this meeting, as much as he's been cited by many different researchers in their talks, as well as being showcased by the Stem Cell podcast. A delightful chat with him and Candice Liu. Please check it out when it comes out. And if you're listening to this late, go back and listen. So fun, that chat. But getting back to Jacob Hanna here. So he used the alternative conditions media to generate these cells that are poised for all lineages effectively. And the upshot there being that you can put them in a dish, just as, these poised cells, alternative condition media cells, which originally arrived in these 4C conditions. So there's other factors in terms of their starting point, but you just put them in and they could form embryo models because they're poised to form, trophectoderm and the Zen and have the pluripotency. So it's kind of, simplifying, again, lowering the barriers to entry for generating these embryo models, but of course, also increasing the efficiency and scalability. So people kind of went crazy in the audience with that. I think a lot of maybe skepticism, but also just confusion in terms of how could these cells be everything and also maintain like a metastable culture, I think was really the question that a lot of people had. But, I think the proof was in the pudding in terms of the capability, and Hanna's very transparent about sharing the data. So there'll be plenty of vetting of that work and soon to be published, I hope, so we can all have a look. Following that, I was really jumping around, so some talks kind of fell through the cracks, but I was able to get in time to hear Wolfgang Wagner from Aachen University in Germany, which was just fascinating for me. He's really out of my kind of range of expertise, but talking about the epigenetic clock effectively and how these age-associated CPGs exist that can be predictive of age. As you age, your methylation is decreased. And so they showed that you could use the CRISPR-based system as kind of an epigenomic editor. And the real fascinating takeaway for me here, because again, I'm naive to the field, is that you can accelerate the epigenetic age of the cells, so make them look older epigenetically by targeting these hypermethylated regions. So like that hypermethylation or the increased methylation that is typical of younger age cells. If you target that, get rid of that, you can make the cells look older. And the converse is kind of true. Because that's what everyone cares about. He showed if you target the hypomethylated sites to try and make them more methylated, you can kind of recapitulate the converse effect, right? Make them look epigenetically younger, but it's a transient effect. So it's not as lasting as that accelerated aging, which the cells stay looking like that for as long as 3 months is when they looked at the distal time points. So I don't know, for me, in terms of the conceptual advance of connecting the epigenome, which everyone talks about, and maybe the intuition of that is there, the link between that epigenome and aging. But here is showing really clearly with the data that it exists. And also, I think the more exciting thing that everyone was at the tip of their seats about was the idea of modulating this, although maybe far-fetched in terms of rejuvenating us as a concept. I think it's at least interesting to consider. Along the same lines of that rejuvenating factor, and the next talk from a Travel Award winner, Lily Guo, was about GDF11, which is a kind of a callback from about a decade ago when Amy Wagers and others identified GDF11 as a circulating factor here in this parabiosis type, vampire equivalent experiment for rejuvenation. But in this case, Lily Guo, who was from Nagy Lab, was using a kind of a cell-based GDF11 delivery device based on ES cells, so engineering the ES cells so they could have induced expression of the GDF11 and then differentiating them into these alveolar cells, so alveolar lung progenitor cells, and then transplanting those, getting them to engraft, and in a fibrosis model, and showing that it resolved fibrosis. So I thought it was a clever, very straightforward approach. And again, the balance in this session, I enjoyed. I caught the back end of this session. I liked how they integrated these, Travel Award who were coming, I don't want to say basic or straightforward. I think that's doing a disservice to the elegance of the studies, but working with systems that were a straightforward idea at least, and showing that in fact they do have this clinical power. And that to me was balanced by Thomas Rando, who came with a deep conceptual, for me, novelty, talking about his lab. And this was a late addition, by the way. He was filling in, but he kind of blew me away with this story about skeletal muscles and how he's shown long ago that there's a spectrum of quiescence from this deep to shallow quiescence. But here he kind of put it in context in terms of the balance of quiescence versus or survival versus reproduction as like kind of a meta idea in nature, and showing that there's a difference here between the resilience of these cells and their proliferation capacity and repair capacity. And I'm not going to go into it deep, but he showed with these really elegant models and knockout mice that this NDRG1 factor, which is like actually a tumor suppressor, that it exerts its effect. Actually, in younger mice, it's important to balance this resilience in order to maintain the stem cell pool. Not just maintain them, but maintain like their capacity and their resilience. How they'll stay alive during the life phase and still be able to reconstitute the muscle. So I thought that was a great conceptual hypothesis and demonstration there, because typically the balance is always between proliferation and like tumor formation. So it was nice for me to have a new idea thrown into the mix there. And then moving on to the plenaries, that started with James Hudson, who's been on the show, friend of the show. He was talking about this massive effort and platform, effectively, that he calls a cardiopedia, which had a lot of moving parts, but effectively it came down to optimized differentiation protocol for making human cardiac organoids that then he was able to scale up and use automation to increase the throughput for screening and use it for this like single, what he called a single human cardiac organoid RNA-seq platform, where they have each of these single cardiac organoids in these arrays under different conditions submitted for bulk RNA-seq and as a platform for discovery. So it has a paper coming out very soon establishing and defining, validating this platform. But I think that what comes of it is going to be more interesting, and I'll be looking forward to that. We've got to get him back on the show. And then we had kind of Duke adjacent, the Duke Medical School in Singapore, Hong Yang Wang, was talking about the reactivation of neural stem cells. And again, a kind of idea on the spectrum of quiescence. As I was talking about the Thomas Rando talking about this is quiescence versus reactivation using Drosophila as a model, showed this really beautiful visual video live imaging model for looking at neural injury and the regeneration from that in these embryonic Drosophila brains. And the real idea here, which I thought was cool, was elevating these microproteins, which are these open reading frames that encode amino acid sequences that aren't appreciated the bioactivity. They're annotated, but they haven't really been described. And what I thought was awesome about this is a graduate student in her lab got to name a gene. I mean, who gets to name genes anymore? But in the Drosophila, they're still doing it. They're still being creative. These genes were named, these microproteins were named SIMBA1 and 2. And the bottom line shown they triggered neural stem cell reactivation. No coincidence by way of Wnt signaling, but the nature of that Wnt signaling I think was important, and the relationship of the blood-brain barrier cells to the actual, I think it was glia or something, but like the microprotein component mechanistically really made sense in the context of the experimental approach taken by Dr. Wang there. So not surprised, Duke, we do it right. But that for me was emblematic of the conference in general. A little bit about new tools, a little bit about novel, exerting the advanced technology to make novel observations and putting it together to identify new genes, new processes. Who would have thought at this point in the game that we're still finding things that are brand new? What about you, Arun? What do you love about the plenary?
Honestly, the highlight for me was the one that came next was Paola Arlotta, who's of course from Harvard. She's been on the show, recipient of the ISSCR Momentum Award, and this was some heavy-duty longitudinal culture of brain organoids and growing brain organoids. Forget this, up to 7 years. This is unpublished data, and I'm sure it's going to be a really high-profile paper. They grew these brain organoids for 7 years, did a bunch of single cell, and then, of course, compared them to the real deal, the human data from the early human brain. They're able to maintain and mature excitatory neurons in these long-term cultured organoid cultures. And it's sort of similar to what happens in the heart and the cardiac organoids. You just had to, make sure they're spontaneously firing. Their functionality and repeated functionality enhances their maturity. That just is very similar to what happens on the cardiac side of things. And part of the reason that they're able to maintain this culture and maintain this spontaneous activity was they used a custom activity permissive media, which I think was building on some of the media that Rusty Gage built many, many years ago. They did long-term, like I said, long-term longitudinal analyses of these organoids, also showed enhanced epigenetic stability over 5 years in culture, and then wrapped things up with a really cool experiment where the Arlotta lab fused the organoids that were young and also old organoids and showed that the older progenitors still made older, quote, more mature cells. So the way she termed it was like an acceleration of their differentiation potential. They basically skipped over some of their younger stages. That was just a lot of questions there, a lot of food for thought. But bottom line was growing. Can you imagine growing organoids for almost a decade? I mean, I'm sure there's gonna be other papers in the similar area on the cardiac side of things, but huge shout out to the Arlotta Lab and Paola for this incredible work, and just one of many papers that's going to be, at the very top of the field for sure. Moving on to the next paper, which was our next presenter, which is Toshihiro Suda, the recipient of the Ernst McCullough Award and the Ernst McCullough Lecture. High mitochondrial mass characterizes enhanced self-renewal in aged hematopoietic stem cells. A really neat observation from apparently the scientific grandson of Ernst McCullough, who was, Dr. Suda's mentor, PhD advisor, actually trained under Ernst McCullough, which is kind of cool. You never know what your lineage might be, right? This mitochondrial high observation was really interesting because these mito high HSCs have a higher proliferation potential. The transplantation rates of the mito high and low are pretty similar, but the mito high cells have a longer sustainability in the bone marrow. So maybe there's some tricks that are waiting to be unraveled there to keep the HSCs longer, maintain them longer in the bone marrow, and ultimately, of course, we're looking towards the goal of sustainable HSC culture and transplantation and indefinite culture, right? So maybe this mito high trick is a way to get there. I moved on into the concurrence and went to a concurrent that was chaired by my boss, Clive Svensson, as well as Malin Parmer, who's also been on the show. This concurrent was really cool. It was stem cell models of tissue injury and repair, and some of our favorite model systems were being represented here, including organoids, as well as the sponge of all things, and I'll get to that in a couple minutes. Started things off with a great talk from Sumra Bayan from the Gurdon Institute in the UK, understanding and harnessing the regenerative potential of the brain with the focus on the cerebellum, which doesn't get a ton of love apparently. It's all about the cerebrum and the cortical side of things, but these are cerebellar organoids that her lab was able to establish. And they were using this as a model to understand cerebellar injury. And apparently the developing cerebellum has remarkable regenerative capacity, and they're able to model this in an organoid system, figuring out how the ASCL1 gene network actually regulates this process in human cerebellar organoids that can also recapitulate these regenerative processes. Next talk was from Deepan, which is a really, really fun talk focusing on the sponge regeneration from Australian National University that was decoding the regenerative landscape, that was providing insights from the sponge of all things. A lot of single cell transcriptomics, and honestly just a really funny talk with a lot of great visuals and pictures of SpongeBob being chopped up, which is interesting. Clive apparently is a big fan of SpongeBob, and he alluded to that in the talk. The sponge has a very strong regenerative capacity, and you can chop the sponge up into many little pieces, and it's going to restore the whole body, just like the planarian flatworm. But the cool thing and the commendable thing about this talk, something we've discussed on the show, when you're using a very unique model system, you have to first develop the tools to actually interrogate the biology of the model system. So the first thing they had to do was develop a single cell atlas of the calcareous sponge, and also figure out the genome of the sponge before you can actually do any of the molecular biology. So again, very commendable tool building for this really unique model system of regeneration. Next talk was from Analilia Serna Valverde from the University of Nottingham in the UK, in vitro modeling of the respiratory infections and idiopathic pulmonary fibrosis using immune-competent multi-tissue pluripotent stem cell lung organoids. They had a really good infection model system for the H1N1 flu virus, which I'm sure can also be extended to SARS-CoV-2 and COVID-19. And in her lab, they were able to derive normal and idiopathic pulmonary fibrosis organoids to serve as an infection platform. So that was really cool to see. Next talk was from Gillian Moffat, investigating the crosstalk between microglia and other cellular models using iPSC-derived co-cultures and organoids. This is a microglia-centric talk. You're talking about the brain, of course. And apparently these iPS-derived microglia secrete trophic factors that target neural progenitor cells, and actually it makes sense, right? The microglia are this auxiliary support cell type that we're really trying to figure out the mechanisms for in the brain, and apparently they can really help out those neural progenitor cells, which can ultimately give rise to new neurons. And also they had a really nice system where they're able to generate myelinating neuroimmune co-cultured organoids. So the theme here was, making these organoids, combining them with other types of cell types, other types of cells to take your models to the next level. And the final talk that I listened to here in this concurrent was from Leondisha Zoldeva. Great talk from the Gladstone Institute in the US in San Francisco, developing a human spinal cord model and showing how spinal cord interneurons can repair the injured spinal cord through synaptic integration. Basically, we're deriving this custom iPS-derived V2A neurons, which had like a really nice capability of regenerating the injured spinal cord in an animal model. Also coupled that with some epigenetics to further control the activity of these iPS-derived V2A neurons. Like I said, her lab transplanted these cells into a mouse cervical spinal cord injury model, and the host axons, get this, are able to functionally innervate into the transplanted cells. So I think a lot of translational potential there. So that was the theme of that session, was, taking the organoids, of course, making the models, making the organoids, and then applying them for clinical applications and diseases such as pulmonary fibrosis or spinal cord injury. You're just, using multiple tools to get to a potentially a clinical endpoint there. Really cool to see.
Yeah, name of the game at this meeting is the clinical endpoint, and that was in abundance. But there was also a lot, as you might expect, when you talk about all these embryo models that are surging, just basic studies of development. So I hit the technology to study development session in my afternoon concurrence, and I'll run through it briefly. Because a lot of the work was published, but some not. Nicolas Rouron, of course, friend of the show and originator of the blastoid, he talked a bit about his work in the lead off talk, and extending that to implantation models, which was really exciting. Really had the audience stirred up, and then it went into really deep basic stuff with Antonio Giraldes from Yale, who was showing with this ultrastructural microscopy, this electron microscopy, nano gold, but of chromatin, showing kind of the spatial relationship with the chromatin, the tightness, and found some really counterintuitive things, or things that ran against the dogma, namely this idea that transcription is going to be enriched in places where the chromatin is looser, right? Which is more accessible. That's what the ATAC tells us. One of the big takeaways with this really amazingly high resolution imaging going down to the level of the nucleosome, you can imagine, and then pulling out in the whole cell. We're talking about millions of these things, showing all the connections. what he showed is that the transcription by his observations was actually enriched in the more tight nucleosome density. So point being is that right there off the bat, you're going against expectations. So a lot of potential for discovery there with transcription under very many different contexts and a lot of different cells, specifically in development. He was looking at the first differentiation event between trophectoderm and inner cell mass. So very pertinent to early development, but cell biology writ large. And there was Maria Jovana Garone from Silvia Velasco's lab at Murdoch's Children Research Institute in Australia, looking at hippocampal differentiation, these really sophisticated long-term diffs of organoids, which were, more specialized, more advanced in terms of their maturation, more faithful to the real thing, benchmarked against true fetal brains. So that was really exciting. And that was across the board, I would say, at this meeting, is showing these more advanced, not just getting farther along, but based on a rationally designed diff program where you're going anterior posterior, where you're looking at the Hox code, where you're starting with the right material. I think that was something that really resonated across many talks, is you got to start with the right material, and you got to very carefully and faithfully track these cells along their path. To that point, some basic understanding of early gastrula embryos, right? That's a key benchmark for doing any of the differentiations that we hope to do to find the bona fides in real embryos. And while human embryos have come a long way, and a lot of data has been presented with human gastrula stage embryos, Jiaping Tan from Westlake University shared spatial transcriptomic data from non-human primates, macaques, and showed and validated the data. It was beautiful to see multiple sections across multiple early Carnegie stages. I think focused on Carnegie stage 9 and 10, and showing how these things could be informative, looking at the germ cell compartment, generating support for this idea of like a new organizer potentially in the allantois, showing like floor plate induction, all things that are really relevant and can be translated to how we conduct our differentiations, right? Understanding that spatial component is going to be a huge part of discovery moving forward in the next few years. So I love that story. And then we ran off. I had to cut that session a bit short to get ready for our delegate recordings, which were also really a lot of fun. I hope you guys will listen to that episode as well. But for now, that brings us to the end of this latest ISSCR 2025 daily recap. Don't forget to follow us on X at STEMCELL Podcast or Blue Sky at STEMCELLpodcast.com to find out what we're up to at the meeting. Also, check back here tomorrow for our final recap of the meeting. That's going to be day 4. See you then.